I wish I had more than one thingy over here. Okay, let's get started with chapter 2. Chapter 2 deals with cells. The whole thing about chapter 2 is cells. Cells are going to be your best friend. So just a little bit of an overview. This is just some historical information that you may want to know. In the early 1600s, Robert Hooke made some discoveries that, hey, there's these tiny little things that might be useful for our lives. We've seen them under these very rudimentary microscopes, and we're not entirely sure what it is. But our boy Robert Hooke over here was like, I think that those things are something. But what can you really tell about cells in 1600 with a non-light microscope? Lights barely even, you had a flame at that point in time, right? Okay. So in about the mid to late 1800s, our two buddies, Matthias and Theodore, discovered some very interesting cells, and they said, hey, I think that these things, these blocks, might be what builds up life. Which was super different from the spontaneous generation theory that we'd kind of been running, I say we as like human society, had been running on for quite some time, right? We just kind of thought that life arose. We weren't sure how, we weren't sure where, we weren't sure why, but we just knew that it did. And our boys here, Theodore and Matthias, were like, hey, maybe, just maybe, these cells have something to do with the growth of that life. Okay? And then your boy Rudolf Virchow, he also does some important things in developing our sense of cells as we know them today. Read about him. Your boy Rudolf. Okay? So what we know about that, cells, is that cells are the smallest living unit in the body. Okay? They are the smallest. A cell's job is to obtain nutrients, make the molecules needed to survive from those nutrients, dispose of waste, maintain their shape, as well as to replicate. A cell is not a cell if it cannot do the majority of these things. Within each cell, we have what we call organelles. Organelles are these tiny subunits within the cells that each have a specific function. Remember earlier today when we were thinking about working in projects, I asked you to think about delegating. Organelles are essentially the delegated tasks or processes that help the overall cell do its job, being obtaining nutrients, replicating, making molecules from those nutrients, etc. Okay? Not all cells will have the same organelles in the same abundance, right? Some cells might be missing. For example, your red blood cells don't have a nucleus, right? They also don't have any mitochondria. So different cells will have different organelles in different abundances, but your general cell will have the majority of the organelles that we'll talk about in a second. Okay? So cells, as we know of, have three main components to them. First, the outer boundary, that is called the plasma membrane. Next is the cytoplasm. The cytoplasm is that kind of inner jelly-like substance within our cells. And we have the nucleus. The nucleus is, for lack of a better, oh, that turned ugly. For lack of a better term, is the brain of our cells. Okay? So, this is just the general structure of a cell, of a general cell. You've seen these. Don't let this scare you. If this bit here is that big center part, what might that be? We just talked about it. The nucleus, yeah. That's the brain of our cell there. This little guy here, that's a mitochondria. Hell yeah, it is. We're going to talk about that in a second. Okay? And all the other little bits and bobs and different organelles that we find throughout a cell. So, the plasma membrane. Plasma membrane is basically a wall that is made up of phospholipids. We call it a phospholipid bilayer. Right? When I was learning this, my teacher told us to throw up your gang signs when you're talking about plasma membrane. And it looks just like this. If this is the head of that polar molecule, these are the tails. And it flips just like that. So that plasma membrane has a hydrophobic head and hydrophilic tails. And then they kind of go butt to butt like that. So when we're talking about plasma membranes, you'll throw up your little gang signs like this. Okay? And that's the plasma membrane is what separates the insides of your cells from the outsides of your cells. Keeps the insides inside and the outside out. The fluid mosaic model is just that lipid bilayer that I was just talking about up there. Okay? In that membrane, you'll find different proteins that help to, one, stabilize, and two, move substances in and out of your cells. Okay? The two types of membrane proteins that we're going to really talk about are integral proteins and peripheral proteins. Okay? Integral proteins are hanging out right on top of the phospholipid layer. No, I'm sorry. Peripheral proteins are hanging out right on one edge, one side or the other. And then the integral proteins are usually embedded inside the membrane itself. Okay? You with me? Cool. So those peripheral proteins help support the plasma membrane from the cytoplasm side. That's the side inside, the jelly side. But there can be some peripheral proteins on the outside. We just call them something different. At that point, they're receptor proteins, but we'll get into that later. You can't see that yet. Don't let this scare you. This is just that phospholipid layer that I was talking about, okay? You can see, oh no, that green is so light. You can see all the little heads of those little phospholipids and the tails, okay? So this protein down here, what might that be? Is it peripheral or integral? Peripheral. It's on the sides. If you guys remember that root peri, means around, right? So those peripheral proteins are probably hanging out around the border, either on the inside or the outside. The integral, that root inter, means, is that in chapter 1 or is that in chapter 2? It's in chapter 2. Inter means between or within. So an integral protein is going to be hanging out right in the layers themselves, okay? So these ones would be integral. Right up in here, okay? Cool? The main function of the plasma membrane, like I said, is to keep the insides inside and the outsides out. Okay? It provides a barrier against substances outside the cell. Some receptors will act as, like I said, receptors to receive information or other things from outside the cell. Say I had you can't see that for squat. A receptor here that was ready to take a little circular molecule right in there, right? So you can have proteins on the outside, sort of like a peripheral protein, that act as a receptor for other molecules, and that will cause downstream effects later in the cell. And that's actually what I study. I'm a molecular biologist by trade, so the processes of something happens on the outside of the cell, what does that do internally to the genetics, to all of that stuff, that's kind of my focus, okay? The plasma membrane gets to determine what substances come in or what substances go out. We call that selective permeability. Permeability. If you, I know dads are really guilty of this, of having very selective hearing. They hear what they want to hear and not much else. That's a very broad generalization. We love dads, okay? But that selective, sometimes I hear what I want, sometimes I'll let in what I want, sometimes I won't. That's that selective permeability that we talk about with the plasma membrane, okay? Let's see if this video will play. Hang on, guys, my computer froze.
The plasma membrane is selectively permeable. It lets some substances through and not others. The plasma membrane is selectively permeable. It lets some substances through and not others. The hydrophobic interior of the bilayer is one reason membranes are selectively permeable. Hydrophobic molecules, those that are soluble in lipids, can easily pass through the membrane.
The bilayer is one reason membranes are selectively permeable. Hydrophobic molecules, those that are soluble in lipids, can easily pass through the membrane.
In addition, small molecules like O2 can sneak between the phospholipids of the membrane. On the other hand, large hydrophilic molecules like glucose and ions such as sodium ions and hydrogen ions cannot pass through the membrane unaided.
Large hydrophilic molecules like glucose and ions such as sodium ions and hydrogen ions cannot pass through the membrane unaided.
Okay, so some things can pass through, some things can't. The bigger the thing, the more likely it will be able to have some trouble passing through that membrane, okay? But our cells have already thought about a way to do. Okay, yeah, that process is called diffusion. So we have a few different types of diffusion. Simple diffusion. Hello? Thank you. Simple diffusion is the tendency of a molecule to move down its concentration gradient, okay? Osmosis is just diffusion of water specifically. That diffusion looks kind of like this. So say I had a box full of circles, box full of oranges, right? One side has all of my oranges. And that's not exactly what I want. I want both sides to have equal amounts of oranges. Not I. Your body wants equal amounts of oranges on both sides. That's called homeostasis. Most of what your body does is in an effort to maintain homeostasis. Homeostasis is just equal balance of everything. So in order for me to move these oranges from one side to the other, they have to pass through my membrane. Thankfully, my oranges are teeny, teeny, tiny, so they can just swoop, move on through until I have both sides with equal quantities of oranges. Okay? That's simple diffusion, going from a high concentration to a low concentration to make them equal. Okay? Osmosis is the same process, but with water. So let's say all of my oranges are water molecules, right? And the oranges on this side are actually salt. So this side, let's flip that actually. With water, you'll go from a high concentration of solute, of a bunch of stuff, to an equal concentration of water and stuff. Okay? That's tonicity. That's a different thing. Osmosis is just simple diffusion with water. Let me not get too ahead of myself. Okay? Cool. So, this image that we're seeing here, this one is simple diffusion, going from a high concentration to a lower concentration, just kind of moving them down the gradient. Okay? Osmosis, moving water through a gradient. The next two are facilitated and active transport. And we'll talk about those in the next slide. Okay? So sometimes the molecules are too big, right? With these molecules here, these lipid-soluble ones, they were small enough to pass through our phospholipid bilayer. They didn't upset the layer too much. Some things will be just too big, too hydrophilic, too not what the membrane wants. So in order to move, but they still need to be inside the cell. In order to move them inside the cell, that's where those integral proteins play a part. That's where they come in. Facilitated diffusion is movement of a molecule down its concentration through a protein. What is that protein again? Integral, an integral protein. Okay? And then active transport is using an integral protein to move a molecule through across the plasma membrane against its concentration gradient. Yeah. I can't spell. Geez. Against its concentration gradient. What does that mean? That means instead of going from a place of low concentration to high concentration, we are taking something from that low concentration and making it even higher of a concentration. That requires active transport, and that requires ATP. ATP is our body's way of making, storing, and using energy. Right? So simple diffusion, osmosis, they don't require any energy. That's just making things happen. Active transport actively needs some energy to throw that molecule from low concentration to a high concentration. Does that make sense? Okay, cool. So there are different ways of molecules, substances entering and exiting cells. One of the ways that we talk about is endocytosis. For those of you that were in lab this morning, you heard me say endocardium is the inner, the within, right? So what do you think endocytosis might be? Yeah, getting something inside the cell is endocytosis. Mechanisms that we use for endocytosis, we have phagocytosis and pinocytosis. Phagocytosis is just the cell eating things. There's a good little bit of carbs right around the corner of your cell. Your cell will go whoop, and just kind of take it and say it's mine now, and then use that information for its own self. Okay? Pinocytosis is just with liquids. It's the same. I'm going to take that. That's mine now, but with liquids. So your cells get a little Pinot Grigio going. They like to drink. That's cell drinking. That Pinot means to drink. Okay? Phago means to eat. And then there's also receptor-mediated endocytosis. Remember how I talked about some cells will have receptors on them, ready for something to bind to it? That is true, right? Sometimes something will bind to it, and it will force the cell to envelop something else. It kind of makes like a bubble for it to pull the nonsense into itself. Does that kind of make sense? Becomes that coated vesicle. This is that. And then forces that substance inside of the cell. Okay? But that only occurs when the receptor has been activated. Okay? That's that receptor-mediated part of it. Because it's very similar to the phagocytosis, pinocytosis of like whoop, enveloping the thing. But this one only happens if the receptor has been activated. Okay? I talked about them. Phagocytosis, take your thing, eat it. Pinocytosis, drink your thing, take your thing, drink it. And receptor-mediated, it has a receptor. Exocytosis, okay? That is the mechanism that our cells use to move things out of our cells. The substance is usually enclosed in a vesicle, and then that vesicle migrates to where? Where might it migrate so that it can leave the cell? The plasma membrane. It goes, because we're still talking about inside the cell here, right? We have stuff in here that needs to go out. It has to pass that plasma membrane. Okay? So that's exocytosis. Exocytosis is a whole process. We're not going to get too into the details of exocytosis. I'll leave that for your molecular biologist. I don't even know what I'm saying anymore, guys.